So I hope that didn't like close you in the middle of a sentence or something like this. But I realized suddenly, oh, it's late. So yes, let's add a little layer to all this. And now we're going to talk about multi-point confocals and light sheet systems. So I'm going to share my screen deck. Yes. Right. All right. Okay, so far we have seen all this and then this. Oh, it's a little bit complex. How do single point confocal work and wide field system? And now we're going to talk about this. And then we will practice all this afternoon. Multi-point confocal and light sheet. So a spinning disk confocal is a confocal that works with a camera instead of working with detectors. So here we have the same thing. We have a sample, we put it on the stage, and then we have the objective. It's an inverted microscope. So the objective is pointing upwards and it's below the stage. And now we have no light. So the sample, everything is dark. But then we come in with our lasers. And on the spinning disk, we use lasers. And the lasers are monochromatic, so we do not need an excitation filter. And so the laser light will meet the dichroic mirror, but you can see the laser light is not focused like a point as it is on a single point confocal. So now it's defocused excitation that meets the dichroic mirror. And now we put a big disk that spins, that rotates very fast in the light path. And what you can see on this disk is that we have all these little holes that are organized like spirals, concentrical spirals. And this disk is spinning super fast in the light path. And so so this is it spins, so it's a scanner. It scans the sample with the excitation light. So it is a scanner. So we have all these through all these pinholes. We have a lots of beams of excitation light that come to the sample, and that makes lots of little dots of excitation, exactly the same as a single point confocal, except just it's a many dots instead of having just one dot. So we're scanning this, and we swipe this on the sample. And so what happens is that the sample is excited, and then the sample starts fluorescing in red because we have green excitation light. And then this light goes back again on the spinning disk and goes through the pinholes, which do the same thing that they do in a single point confocal. So they get rid of the autofocus light. Okay, so again, we have here the objective focal plane, and here we have the focal plane with pinholes. And so this is the reason why we can say that this is a confocal. It has two conjugated focal planes. And so it's a confocal, a real confocal. And then what happens is that all this excitation light goes through the pinhole, is rejected, it is reflected by the dichroic mirror, and it goes through the emission filter. And now we're scanning this onto the camera very, very, very fast. So we can record actually with a camera because we have so many little spots, and we are scanning it on the camera. And then we get an image. It's a digital image, so it has lots of pixels. And if we look at each pixel, we can see that each of them has an XY coordinate and an intensity. So it's a confocal, but it's very fast because we acquire lots of spots at the same time. And we illuminate many X-wide spots. And the illumination is like small cylinders in Z. So clearly, we also illuminate in several Z planes. But when all these spots of illuminations are sending some emission, the autofocus light is eliminated by the pinholes. And then the camera is the device that records both the intensity and the XY information that we find in the image. So both the excitation line goes through the pinhole and the emission light also goes through the pinhole. So both of them are scanned. And so how does that work? So this is a little bit a nice schematic by the Zeiss. And sometimes some spinning disk systems have a double disk like this. And the role of the first disk is to focus the light. And then the role of the second is, so it has micro lenses here. And the role of the second, this disk is the pinhole, really, the same thing as in a single point confocal. And here we see this is an upright system because the objective is pointing downwards and is above the sample. So you can see here the laser light coming in. The first disk is focusing it, and it goes through the dichroic mirror, and then through the pinholes, through the objectives onto the sample. The sample fluoresces in many, many spots, and the emission light comes back through the objective because in fluorescence, both the excitation and the emission go through the objective. And I go through the dichroic mirror, and this is reflected towards an emission filter and then a camera with many, many pixels. So how does it work? Why is it that we do not see spots on our image? But that is because the spinning disk spins very, very fast compared to the acquisition time. And so the camera sees through the disk. And this is the same thing a little bit as this little guy. So you can see this very cute little guy there, and he starts spinning and spinning. And when it goes very, very fast, and we're recording this, you see we're missing the lines there. We do not see them anymore because the wheel goes so fast, and then clearly some bad stuff can happen to the little guy in the wheel. But this is the idea. It's the same thing as when you're cycling and then someone goes so fast that you don't see the spokes. So click. So this is the same type of things that happen. It's just a question of how fast the disk rotates compared to the acquisition. So what are the advantages of using a single point confocal compared to a multi-point confocal since they are so much faster, right? Well, the main advantage of a single point confocal is its flexibility. You can be flexible on the scanned region. So you could say, I'm going to be imaging only one line, or I'm going to be imaging only a small spot in my sample. You can scan whatever you want because you're scanning point by point. So you can make a shape and the size of the scanned region as you want. On the camera-based system, like a spinning disk multi-point confocal, we're stuck to the scanned area, and that's a square or a rectangle. We can crop the image afterwards, but we're stuck to that shape. And then in a single point confocal, the scanning, we can scan an area of any shape and size, including a small spot. In a single point confocal, we also have flexibility of the illumination power. On a single point confocal, the laser light is really, really concentrated into a tiny spot. So we have very high power in this small spot. That means we have a lot higher bleaching. So we will be talking about bleaching later in this course. But the light is very concentrated when we use a single point confocal. But this can be very useful for some techniques which need a very high illumination power. For example, FRAP, where we're bleaching or photoactivating a small region of interest, and then afterwards we want to image the whole field of view, but also STED, that we will also talk about this in the super resolution lecture, where we can shape the laser spot to improve the resolution. So we have several people in this course who use STED this year. We also have on the single point confocal the flexibility of the pinhole size. So we can, because we have only one pinhole and it is a diaphragm that we can open and close, it's an iris that we can open and close, then it means we can make this pinhole size perfect for any wavelength and any objective numerical aperture. We will see later what that means to make the pinhole perfect, but just know that it is possible to open and close the pinhole on a single point confocal. On a multi-point confocal, a spinning disk system, it is not possible to open and close. So we have just this disk with lots of holes in it. It's possible to change the whole disk, but we have very little flexibility of adjusting the pinhole size on a multi-point confocal. So the pinhole size is perfect only for one objective. It's designed only for one objective and one wavelength on a spinning disk, multi-point confocal. Another type of flexibility we have with a single point confocal is the pixel size. So on a camera-based system, the pixel size depends on the camera sensor. So we have a fixed pixel size for a given magnification. Whereas on a single point confocal, we can set the pixel size the way we want because the pixel size is the distance that the scanner is jumping inside the sample. So it images one spot and it jumps to the next spot, and that determines the pixel size. So we can change that in the software. So we have a high flexibility for pixel size, and we will see later what the implications of this are in our images. So we can get the perfect size for any wavelength and objective with a single point confocal. So these are the advantages of single point confocal. You can see that all of them are related to flexibility. And of course, we have the flexibility of the emission filter, where we can have virtual filters and spectral detection, and that is also a great advantage of single point confocals. So now they are, sorry, I just wanted to check that I had not forgotten anything. No. So now there are some tricks. We know that the multi-point confocal is very fast and the single point confocal is very flexible. But there are some tricks that can sort of get them to meet a little bit halfway. Let me just close this in here. Yes, so I can see the time and I check. Yeah, we're good. So the first difference between the single point and the multiple confocal is clearly the speed. So how can we make single point confocal faster than they are? So the camera-based system are much faster because they use a camera, but there are some tricks to make a single point confocal faster. We can have bidirectional scanning, and maybe some of you have seen that we can scan bidirectionally on your system or monodirectional. And we will talk about this also later. So we can make it faster. We can use a resonance scanner. If some of you have a resonance scanner on your single point confocals, that goes also faster. We can use image in parallel, so we can image several colors at the same time because we have several detectors. So that also makes us go a little bit faster if we use several detectors. And we can use spectral detectors, which can also image lots of colors at the same time and make it go faster. And then the second difference between single and multi-point confocals was the flexibility. So single point confocals have a high flexibility, but there are some tricks that can help camera-based systems. So for the flexibility of the field of view size, we can crop and not acquire with all the we don't have to acquire with all the pixels of the camera. We can just decide we're going to acquire only with the center, for example. So we cannot choose the shape, but we can make the field of view smaller. We can also have a little flexibility with the pixel size, and that is because we can add an extra magnification that we have sometimes on our microscopes. We can put an extra lens in a system that increases the magnification. We will talk about the connection between magnification and the pixel size later. We can also bin the camera, and that means we can make bigger pixels instead. So we have a little flexibility in changing the pixel size on the camera-based system. And the pinhole size, yes, sometimes we can have a different disk. So we could change the disk, but it's clearly, at the maximum, you're going to get two sizes. It's very little flexibility compared to a single point confocal. And the flexibility of the emission filters, clearly we could have several cameras, and that would allow you to image simultaneously several fluorophores. There's also some tricks where you can image a smaller field of view, different colors on different areas of the camera. But so that is also a possibility to make it more flexible, where we can have different emission filters and combine imaging. But all this we will talk about a bit later in the workshop, the speed versus noise, and also in the imaging multiple colors at once. So now you can see that if you want to image fast, you definitely go for a camera-based system. But if you want to use the flexibility of a single point confocal, that's what you should be using. So the main issue that I have is when people use a single point confocal with a very standard way. For example, whatever they want to do, they're going to use a pinhole of one area unit. They're going to use image at 1024 by 1024 with the full field of view. They will always image sequentially. I think that if you're using a single point confocal and you accept that your imaging is very slow, there must be a reason for it. So if you're not using the flexibility of the single point confocal, you should not be using it. You should be using a multi-point confocal. You would go much faster and get the same type of images. So all this is something that you are going to be discussing, and you will try to figure out if the machine that you're using is actually the best for your experiments or not. These are some of the assignments that you will have. So now that I spoke about the multi-point confocal, which is also called spinning disk, we're going to talk about light sheet. And there I don't have any fancy graphs or anything. I'm just going to show you pictures. Light sheet systems are sort of a little bit the latest on the market, even though this has been invented a very, very long time ago. But some technical advances in cameras and lasers and stuff have made it come back very strongly, and also in clearing techniques. So they can look very, very different. So for example, this can be a light sheet system. This is Z1 from Zeiss. I think some of you have been using it. This is another light sheet system, one that uses 4 different objectives, and it's a flat architecture. Yeah, so as you know, they are very, very different, but the principle is the same, that we always have the excitation and the emission light path are perpendicular to each other. So we have seen that in fluorescence microscopy, both the excitation and the emission go through the objective. On most systems, we use only one objective when we image, but on a light sheet system, we often use at least 2. And so in that case, we have one objective that is going to be for excitation and one for emission. So it's still true that the excitation and the emission go through an objective, but in a light sheet, it's sometimes not the same objective. So the excitation and the emission are perpendicular, and only the plane of interest is illuminated. So we have optical sectioning, but because only the plane of interest is illuminated, the shape of the illumination is like a sheet. So we do not bleach above and below the plane like we do in a single point confocal and multi-point confocal and wide field. We only illuminate the plane of interest, and that's what we're imaging. So this is the light sheet system that we have at our facility, and it uses an upright microscope with 2 objectives at the same time that go and dip into the medium. And this is another example of a schematic by Zeiss. So a light sheet system is a camera-based system. So it is very fast, but has a low flexibility. The excitation and the emission are perpendicular to each other. So the 2 light paths are perpendicular to each other. And that means that only the plane of interest is illuminated. So you can see here the excitation objective. This is the emission objective. And here we have the sample. It's a zebrafish that's hanging in there in between. And what we have in here is that we have a sheet of light, like a knife of light. It's vertical in this schematic that comes through the excited objective and only illuminates like a knife cutting, right? With light. So it only illuminates one plane, and only that plane starts fluorescing. And then we have the emission objective here and the camera behind. And so only this plane will fluoresce. And when we acquire the image, we just acquire like a wide field system. So whatever comes out of the sample, we acquire. We have no pinhole, and we acquire all the photons that are emitted. So there is no emissions from the other planes because we have this sheet of light that just illuminates like a knife. And And this means we have no bleaching and no photo damage from other planes. But we anyway have optical sectioning. So we have a high Z contrast compared to a wide field system. And the emission light path is exactly the same as in wide field. So as exactly like in wide field, whatever photons come out of the sample, we are going to collect them with the emission objective and then send them to the camera. We will not throw them away with a pinhole like we do for single point and multiple confocals. So this is a great advantage of. So now I want to show you an old movie from the Leica light sheet, and I think Leica has stopped making this type of light sheet, but I really like it because it clearly shows how the sheet is formed. So in this particular light sheet, there's only one objective. It's pretty rare. Most light sheet systems have two, four. But anyway, the light path is the same. So the excitation and emission light paths are perpendicular. So let me play this little movie and you will see what happens. So here we have a specimen. It's a C. elegans embryo. And here we have the emission objective, and here we have the excitation objective. So there is um, here we have a sheet of light. So this is what is not normally on most light sheet system, but this was the system that Leica had. They had like a little mirror. And so the sample goes there in the middle, and they had only one objective. So they scan the excitation light, and in here you can see the beam of light that bounces like this and goes back to the objective below. And now they start scanning this, and you start scanning and scanning very, very fast. And again, it's a question of speed. If you scan fast enough, it's going to look like a sheet. And that's exactly what happens in all light sheet systems. So now they're showing as a detail, we have the sheet of light in blue, and then the sample is moving across, and you can see how only the plane where the sheet of light is going to be excited. And this is the only thing that fluoresces. And so whatever photons come out, this is what we are going to image, right? So I think that explains quite nicely how we make the sheet of light by just scanning it very, very fast, and also how only the plane of interest is excited and not the plane above and below. So we have an optical sectioning capacity, but we do not bleach above and below the plane. So what are the challenges? Why is it that we don't all use light sheet microscopy? Well, in general, light sheet microscopes are designed for quite big samples, and so they're not transparent. And we have to do something to make them transparent, and that is something we will discuss during the clearing lecture. And so there is a difficulty in making the sample transparent. It's a little bit of a challenge, let's say. And then it's very restricted because you have two objectives or four objectives, and so the space is restrictive. So sometimes the mounting of the sample is not easy between all these objectives. The sample must also be accessible from both the excitation and the emission side. So it's not always easy to mount the sample. And then any imperfection is going to create a shadow. So that is a typical problem of light sheet microscope. Different manufacturers will find different solutions, like rotating the sample and stuff like this. Ours has an airy beam to correct for that. And also, it's a bit of a problem if you need to fuse. If you have a big sample and you need to acquire from different sides, let's say, and then you need to fuse. It also creates some challenges because you require very big data. And then you get, in most light sheet systems, except the one we have, you get either good Z resolution or a large field of view. You don't get both of them at the same time. And so very often you need to deconvolve as well. And that's not always perfect if the clearing is not perfect. So there are some challenges with sample preparation, let's say, and mounting and handling the data, because in general, we acquire lots of data. So in a light sheet microscope, clearing and matching the refractive index is absolutely crucial. So here we have, why do we use a light sheet system or versus the others? So all three systems, multi-point confocal, single-point confocal, sorry, and light sheet have an optical sectioning capacity. In a single point and multiple confocal, the excitation and the emission go through the same objective, but not in light sheet. They go through perpendicularly. So we have several objectives. In the light recording, the sensor is a camera for single point, is a detector for single point confocal, but it's a camera for multi-point confocal and light sheet. This means that a single point confocal is slow. So we're going to have 0.5 images per second, maybe for a single point confocal, whereas we can go to 100 images per second at full resolution with the camera-based system. We have a very high photon efficiency with light sheet because we don't have pinholes, whereas we have a lower photon efficiency also because we use here detector, which has a lower efficiency, like I mentioned before. And then we have much lower bleaching in the light sheet system compared to the others, but the highest is definitely the single point confocal because the light is concentrated. And then we have excellent flexibility of pixel size and Z-step, and so very high flexibility with a single point confocal, but much lower flexibility with the other two systems, even though we have a very good resolution in XY. So, and here we have to image very deep. We can image very deep in the light sheet system compared to the other two systems, the confocal systems. So there are some advantages and disadvantages for each of them. And the reason why we use one system is not random. We have to pick the system that is best for our scientific question and our sample. And so that's what we are going to do in this course, hopefully. So I just want to say that in the lecture Anatomy of a Microscope, there is now a little document that is available where you have a summary, an annotated summary of all the different light paths. If you have, it's quite nice and visual like this, so you can have a look if you want. So which microscope should you use? To start with, if you can use a wide field system, it's very fast and it's very photon efficient. If it's not possible to use a wide field system because your sample is a little bit too thick, then maybe you want to use a spinning disc confocal. That would be choice number 2. Because a spinning disc confocal is a lot less light efficient because we throw away some light, but it has an optical sectioning capacity and it's anyway super fast. So that's very good. But if you cannot use a spinning disc confocal, then you go to a single point confocal where it's slow, but you have the flexibility. And then if you have a big sample, then you can use a light sheet system or very dim samples as well. That works very well because it's light efficient. So this would be choice number 1, and then number 2 and number 3 and number 4 in that order, if it's possible. But unfortunately, a lot of people choose the single point confocal because they think it's the best and they do not know. So that's what we're going to try and maybe make you realize here during the anatomy of the workshop this afternoon. So in the anatomy of the workshop, we're going to look at 4 light paths, transmitted light in the eyepiece, fluorescence in the eyepiece, imaging transmitted light and imaging fluorescence, and you're going to study the 4 light paths until you get super expert at knowing what is what and what does what. And I think it will become very clear afterwards. So here is the conclusion of this. A multi-point confocal is usually called a spinning disk. They are very fast and they have optical sectioning capacity. However, they have low flexibility, so you cannot pretty much change the pixel size or the pinhole size, and you cannot acquire several colors simultaneously for most systems. And the light sheet systems are very fast and they have optical sectioning capacity, but they are often optimized to image large clear samples, and they have a very high photon efficiency and low bleaching. So they are really nice for a lot of things, light sheet systems. So again, all the animations that I've had are available in Canvas for these lectures. So I'm going to stop in here and ask